Okay, I think we are all set. Okay, so hello everyone. So I'm Kenny Trieu. I'm a PhD candidate here at the University of Michigan in the Cell and Developmental Biology program. And for my talk that I'm going to be giving to you all today is simply just how to grow a skin tumor. This is my PhD thesis research that I'm currently doing here. So before I talk about skin tumor and all of the nitty-gritty details, I first wanted to explain and talk to you all about cancer. So what is cancer? So ultimately, cancer by definition is when our cells in our body start to go out of control and they take over our healthy cells in this context. So here I have schematically shown to you a gray circle here. So think of this gray circle as a cell in our body. So we have this gray cell here. And what happens in cancer is that what happens to this gray cell is that there's a change in the DNA. Some change happens in this DNA in the cell, for better or worse. And what happens is that over time, when you have this change in this cell here, what's going to happen is you're going to start to grow a tumor. And these cancer cells are going to start taking control and go out of control and take over the healthy cells and form a tumor in this context here. But I'll add one bit of context here, a little bit more. And what actually happens in cancer is that there's actually multiple changes in the DNA here. So you see these kind of lightning rods here, and you see there's actually multiple changes that are occurring in this tumor. So really, my thesis research project that I'm working on here at the University of Michigan is I'm very interested in studying exactly what are the changes in the DNA that actually promote skin tumor formation. So, you know, skin cancer, I'm sure a lot of you guys are aware of that. You happen to know maybe a family member that has been affected by it. And particularly the skin cancer that I'm very interested in studying is called basal cell carcinoma, BCC for short. And what BCC is actually, it's actually indeed one of the most common cancers here in North America, where we see about actually 1 million new cases diagnosed each year. And a picture on the left-hand side here is actually a BCC on a human skin here. So it's these little tiny bumps, or it could be very large bumps in certain cases that appear on our skin. So in order to really study the biology and kind of the science behind how the skin tumor grows, I need to have a model in order to study this. And really in my lab, we utilize a lot of mouse models to study skin cancer. So a mouse model is one approach to really find a way to identify and really study the human disease in our setting. So we use a lot of mouse models, and really I want to put in this analogy here is that currently what I'm doing in my lab research is that I'm generating lots of models. So if you think of a model, you can kind of think of it as a car model. So, you know, when I joined the lab prior, we had a first model where we had one change in the DNA. You can kind of refer it to the model T. Very, very preliminary, very initial car back in the past. But when I joined the lab and as I'm currently studying and progressing in my PhD research, I've kind of incorporated a second model, maybe an upgraded model where it's kind of a Volkswagen, where I incorporated 2+ DNA changes in our mouse model. And ultimately, my time here at the University of Michigan is I'm really incorporating a lot of different DNA changes, like I said in the second slide, where there's actually multiple changes in the DNA, and I'm progressively upgrading our different models, kind of to an Audi and ultimately to our Tesla. And when you think of a Tesla, it's got all the Wi-Fi capabilities, it has the touch screen, the auto drive, all the fancy things, and somehow I'm kind of referring it back to these upgraded models where there's multiple changes in the DNA that could perhaps maybe keep a skin tumor to grow. So where do I see this, and what do I want to do with all these models that I'm generating during my time for grad school? Is that I have all these different models, the first one, the second one, the third one, the fourth, and I'm upgrading them with more changes in the DNA. And ultimately, using these mouse models to study the human disease, I want to use it as a platform to have a system to use to study for clinical drug testing. And ultimately, hopefully, these mouse models can really find some way to pinpoint some drug testing to really eradicate skin cancer in this context. So really, I hope that you guys all learn that what I'm doing currently right now is that we're looking at these mouse models. We're trying to study how skin tumor grows, but ultimately, when we use these models, can they be that important tool to really identify these novel drug therapies to really eliminate cancer as a whole? And for that, I want to thank you all for listening to my talk. My email is to the bottom here if you want to email me and if you have any questions, or you can always shoot me up at Twitter. And thanks again for listening. Bye-bye.